Good night, everyone. My name is Vanessa Ramata, and tonight I will be discussing the cerebellum and its coordination pathways. So this is an outline of what we will be covering within this presentation. I will be discussing the gross anatomy of the cerebellum, its morphological subdivisions, the nuclei, cerebellar peduncles with their afferent and efferent connections, the coordination pathways, and some clinical significance. The cerebellum, which was coined the little brain by Leonardo da Vinci in 1501, refers to the largest part of the hindbrain located in the posterior cranial fossa. On average, it weighs about 150 grams. The functions of this system include maintenance of balance and posture, maintenance of tone, coordination of voluntary motor activity, and these include smooth, coordinated muscle movements, rapid muscular movements, control of the rate, range, force, and direction of movement, speech production and eye movements, and also motor learning. Diseases within this system manifest as a triad of motor dysfunctions, which include disequilibrium, that is loss of balance, characterized by gait and truncal ataxia, hypotonia, that is loss of resistance normally offered by muscles on palpation, and dyssynergia. This is loss of coordinated muscle activity. These diagrams show the gross anatomy of the cerebellum. It sits within the posterior cranial fossa, underneath the tough layer of dura mater called the tentorium cerebelli, and behind the pons and the medulla. It is separated from the pons and the medulla by the fourth ventricle. It consists of two large lateral hemispherical lobes, the cerebellar hemispheres, which are united to each other by a narrow median worm-like portion called the vermis. It is connected to the brainstem by three pairs of large fiber tracts called the cerebellar peduncles. The surface contains numerous fissures, separating narrow folia, which increases the surface area of the cerebellum. The surface contains the gray matter, that is the cerebral cortex, the cerebellar cortex, while the white matter under this contains the four cerebellar nuclei embedded within it. There are two fissures which divide the cerebellum into three structural loops. These fissures are the primary fissure, which divides the anterior and posterior lobes, and the posterolateral fissure, which lies on the inferior surface and separates the flocculonodular lobe from the rest of the cerebellum. Based on functional criteria, the cerebellum is divided into three parts: the vestibular cerebellum, or archicerebellum, the spinal cerebellum, or paleocerebellum, and the cerebral cerebellum, or neocerebellum. The vestibular cerebellum is described as the oldest part of the cerebellum. It consists of the flocculonodular lobe and the lingula. Its connections are chiefly vestibular, receiving and sending information from the vestibular nuclei and the visual cortex. It is involved in the control of balance and ocular reflexes. The spinal cerebellum consists of the anterior lobe and the pyramid and the uvula of the vermis. It receives proprioceptive information from the dorsal column of the spinal cord. Its connections are concerned with tone, posture, and coordination of movement. The cerebral cerebellum consists of the middle lobe, the largest part of the cerebellum. It is chiefly cortico-pontocerebellar. That is, it receives signals from the cerebral cortex and pontine nuclei. It sends out signals to the thalamus and the red nucleus. It has roles in motor learning, coordination of muscle activation, and visually guided movements. This is the most recent part to develop and is most prominent in higher mammals. The internal structure is very intricate. The cerebellar cortex is a thin surface layer of gray matter. There's a central core of white matter with masses of gray matter embedded within it, which are the intracerebellar nuclei. The cortex consists of three distinct layers, which are the outer molecular layer, an intermediate Purkinje layer, and an inner granular layer. At the molecular layer, we can see here that the Purkinje fibers have fine branches at the end of the nerve fibers which extend into this area. This is referred to as the dendritic arborization of the Purkinje cells at this layer. There are a few and widely spaced nerve cells, which are the stellate and the basket cells. Within the white matter of the cerebellum, there are intracerebellar nuclei. These are masses of gray matter embedded within and are noted to be 4 in number on each side of the midline. From lateral to medial, these are the dentate nucleus, the emboliform nucleus, the globose nucleus, and the fastigial nucleus. The dentate nucleus is the most prominent of the nuclei, the largest in primates, notably in humans. It is the nucleus of the neocerebellum and therefore receives afferent fibers from it. The efferent fibers leave via the hilum facing anteromedially, with these fibers forming most of the superior cerebellar peduncle. They include the dentorubral and dentothalamic fibers. That is, they relate to the red nucleus and the ventral lateral nucleus to the thalamus. Fibers from these then project to the spinal cord and the cerebral cortex. The emboliform and the globose nuclei together are referred to as nucleus interpositus. They are the nuclei of the paleocerebellum, receiving afferents from it and send efferent fibers to the contralateral red nucleus. From there, its fibers project to the spinal cord by the rubrospinal tract. This facilitates flexor muscle tone. The fastigial nucleus lays near the midline in the venous. It is the nucleus of the archaeocerebellum, hence receives afferents from the flocculonodular lobe and conveys efferents to the vestibular and the reticular nuclei. Fastigial connections influence extensor muscle tone. The white matter is made up of 3 types of fibers: the intrinsic, the afferent, and the efferent fibers. The intrinsic fibers remain confined within the cerebellum, connecting different regions within the same hemisphere or the 2 hemispheres. The afferent and efferent fibers are described before, connect the cerebellum to other parts of the CNS. The afferent fibers carries inputs from the cerebral cortex, the spinal cord, and the vestibular apparatus, the red nucleus, and the tectum of the midbrain. Information is carried from the cerebral cortex to the cerebellum via the cortico-pontocerebellar, the cerebro-olivocerebellar, and the cerebro-reticulocerebellar pathways. The spinal cord sends proprioceptive information via the posterior spinocerebellar, anterior spinocerebellar, and the cuneo-cerebellar tracts. The fibers from the vestibular apparatus pass to the cerebellum either directly or after relay in the vestibular nuclei. These fibers enter the cerebellum mainly through the inferior and middle cerebellar peduncles. The fibers are classified into 2 types. So they are climbing fibers and mossy fibers. Mossy fibers receive input from the spinal cord, cortex, and the brainstem, therefore constituting the bulk of the afferent fibers of the cerebellum. Each fiber ends in 30 to 40 terminal swellings called a rosette. They form a synaptic contact with dendrites of the granule cells and a few Golgi cells, resulting in the formation of a cerebellar glomerulus. It is spherical and is surrounded by a capsule. Therefore, it can be seen that one mossy fiber synapses with several granule cells. And then each granule cell synapses with thousands of Purkinje cells via the parallel fibers. On the other hand, climbing fibers receive inputs mostly from the inferior olivary nucleus. Each fiber gives a collateral to the intracerebellar nucleus, then makes monosynaptic contact with a Purkinje cell. This summarizes the pathway of the afferent fibers to the cerebellum, and now we'll talk about the efferent fibers. The efferent fibers leave the cerebellum carrying output signals to the red nucleus, thalamus, vestibular complex, and the reticular formation. The output of the cerebellar cortex is through the axons of the Purkinje cells. Most of these axons end by synapsing with the neurons of the intracerebellar nuclei, which then project to other parts of the nervous system. A few of the neurons from the flocculonodular lobe and vermis pass directly out of the cerebellum to end in the lateral vestibular nucleus. Fibers from the dentate, emboliform, and globus nuclei leave the cerebellum through the superior cerebellar peduncle. Those from the fastigial nucleus leave through the inferior cerebellar peduncle. The intrinsic neuronal circuit of the cerebellum is complex. All of the afferent fibers, that is the mossy and climbing fibers, are excitatory to the cells of the cerebellar cortex, shown here, and the deep cerebellar nuclei. On the other hand, all of the cells of the cerebellar cortex are inhibitory, except the granule cells, which are excitatory. Just to recap, the climbing fibers are the fibers which are monosynaptic with Purkinje cells, and these bring proprioceptive stimuli from the inferior olives via the inferior peduncles, sending some fibers to the nuclei, and the rest of fibers to excite the Purkinje cell when the neurotransmitter aspartate is released. When the Purkinje cell is excited, it then releases the neurotransmitter GABA to inhibit the deep cerebellar nuclei. This controls the overshooting and undershooting of movements. The mossy fibers, on the other hand, has multiple synapses. These neurons bring signals from the sensory pathways, giving off some fibers to the deep nuclei, which is stimulated when glutamate is released. Some fibers continue on to form excitatory synaptic connections with the dendrites of the granule cells, forming what we call the glomeruli, and with Golgi cells. The granule cell axons ascend upwards into the molecular layer, spreading out to form the parallel fibers. The parallel fibers stimulate Purkinje cells, stellate cells, and basket cells. Both stellate cells and basket cells, when stimulated, release inhibitory neurotransmitters at the Purkinje cells. This is referred to as a neural sharpening mechanism. Therefore, in executing precise motor movements, first, a rapid excitatory signal from the deep nuclei to the motor output pathway enhances motor movement. And then this is followed within another fraction of a second by an inhibitory signal. This inhibitory signal produces a damping effect. That is, when the motor system is excited, a negative feedback signal occurs after a short delay to stop the muscle movement from overshooting its mark. Otherwise, oscillation of the movement would occur. So what is a cerebellar peduncle? This refers to the grouping of the afferent and efferent fibers on each side into three large bundles called the superior, middle, and inferior cerebellar peduncles. The superior cerebellar peduncle emerges from the anterior cerebellar notch, runs superiorly to enter the lowest part of the midbrain. It is the main efferent pathway from the cerebellum, and its fibers arise in the dentate nucleus. The middle cerebellar peduncle is the largest of the three peduncles, arising from the pons, and becomes continuous with the white matter of the cerebellar hemisphere. It consists of only afferent fibers from pontine nuclei to the opposite cerebellar hemisphere. These are called the pontocerebellar fibers. The inferior cerebellar peduncle consists of afferent fibers to the cerebellum from the spinal cord, olivary nuclei in the reticular formation of the medulla, and the vestibular nuclei and nerve. It also has efferent fibers from the cerebellum to the medulla. That is the vestibular nuclei and the reticular formation. Now I'd like to go into a little more detail with respect to the pathways or fibers passing within each cerebellar peduncle. As we saw before, the superior cerebellar peduncle has both afferent and efferent tracts. The efferent connections here extend from the deep nuclei to the brainstem or the thalamus. An example of this is the dentorubral, dentothalamic, and dentorubral-thalamic pathways. The dentate nucleus sends signals via the axons to the contralateral red nucleus within the midbrain and to the contralateral thalamus, which then sends signals to the cortex to form a motor plan or response. If the red nucleus has been activated, it crosses to the contralateral side and moves down to the spinal cord to the flexor muscles of the extremities. This is the rubrospinal pathway. The afferent tracts passing through this peduncle includes the ventral spinocerebellar tract, which takes proprioceptive information from below L2 and 3. The rostrocerebellar tract, which takes similar information from the cervical region. The tectocerebellar tract, which takes information from the tectum. This refers to visual stimuli from the superior colliculus and auditory stimulus from the inferior colliculus. This helps to send information to the cerebellum to control ocular movements and head movements to the stimuli. The middle cerebellar peduncle consists of only afferent fibers. The afferent tracts include the pontocerebellar tract, ceratogenic fibers, and the reticulocerebellar tract. The pontocerebellar fibers form the bulk of this peduncle, with fibers arising from the pontine nuclei, running transversely across the midline and entering the contralateral posterior lobe of the cerebellum. The pontine nuclei receive input from the cerebellar cortex, thus forming cortico-pontocerebellar pathways. This is the largest pathway of the cerebellum and relays motor and sensory information from the cerebrum to the cerebellum to coordinate voluntary movement, gait, and fine motor control via the efferent pathways. Lastly, we have the inferior cerebellar peduncle. It is primarily afferent, but also contains some efferent pathways. The afferent pathways include the dorsal or posterior spinocerebellar tract. This takes proprioceptive information from C8 to L203. They arise from the ipsilateral thoracic nucleus going to the ipsilateral cerebellum. The cuneo-cerebellar tract takes proprioceptive information from above C8 via the dorsal gray horn to the ipsilateral cuneate nucleus, then via the external arcuate fibers to the cerebellum. The vestibulocerebellar tract receives information from the inner ear to the vestibular nuclei and into the inferior cerebellar peduncles. The olivocerebellar tract takes proprioceptive information and it's carried to the spino-olivary tract to the contralateral olivary nucleus, then crosses over that level to the contralateral cerebellum. The reticulocerebellar tract is all sensory information from the body goes via the reticular nuclei of the medulla to the ipsilateral cerebellum. The main efferent tracts of the inferior cerebellar peduncle include the cerebello-reticular tract and the cerebello-vestibular tract, which isn't shown here. The cerebello-reticular arises from the bilateral fastigial nuclei and goes to the pontine and medullary reticular formation. The medullary area facilitates flexion and the pontine facilitates extension. If they're activated, they send down the information to the spinal cord and activate those muscles. The cerebello-vestibular tract, the information is sent from the Purkinje fibers to the vestibular nuclei and up via the medial longitudinal fasciculus to control extraocular eye movements. And they can move down via the vestibulospinal tract and activate anti-gravity and postural muscles. The cerebellum plays a key role in accomplishing smooth and coordinated movements by means of its comparator function. When the cerebral cortex decides on a movement to perform, its impulses stimulate the anterior horn cells of the spinal cord to initiate voluntary movements. At the same time, the cortex tells the cerebellum about the intended movement, while the cerebellum is also receiving information from the muscles about the information that is about the actual movement that is occurring. The cerebellum then compares the intended movement with the actual movement occurring, and if differences are detected, information goes to the motor cortex and the lower motor neurons to correct the discrepancy. This ensures that movements are accurate in time, rate, range, force, and direction. Therefore, the cerebellum plays a very important role in the learning of motor skills. Now onto the vasculature of blood supply. The cerebellum is supplied by 3 pairs of cerebellar arteries. The superior cerebellar artery, which is a branch of the basilar artery, which supplies the superior surface. The anterior inferior artery, which is another branch of the basilar artery, and it supplies the anterior inferior surface. And the posterior inferior cerebellar artery is a branch of the vertebral artery and supplies the posterior part of the inferior surface. The venous drainage is provided by the superior and inferior cerebellar veins. These veins drain into the superior petrosal, the transverse, and the straight dural sinuses. We'll now discuss the numerous ways in which cerebellar dysfunction may manifest. It is very important to note that the cerebellum receives input from and gives output to the ipsilateral side of the body. Therefore, cerebellar damage or diseases results in deficits of the ipsilateral side. Frequently, these clinical findings are remembered by medical students by using the acronym Danish. So we have dysdiadochokinesia, which is the inability to execute alternative movements in rapid succession. We have ataxia, which is uncoordinated movement. Nystagmus, which is involuntary oscillatory movements of the eyeballs. Intention tremors, which are tremors with purposeful movements, and they disappear with rest. Scanning speech, which is slurred, prolonged, explosive, and has noticeable pauses. And hypotonia, which is decreased muscle toning. The acute onset of cerebellar signs or symptoms are considered an emergency, as it can be due to vascular causes such as stroke or hemorrhage, or it can be due to cerebral edema. Chronic cerebellar symptoms can be due to genetic causes such as Friedreich's ataxia or spinocerebellar ataxia, or due to acquired conditions like chronic alcoholism or brain tumors. Signs of cerebellar lesions or disease can be described as they relate to the affected functional locations within the cerebellum. Lesions involving the vestibular cerebellum produces signs such as truncal ataxia, staggering gait, and nystagmus. On the other hand, lesions involving the cerebral cerebellum leads to muscular incoordination and manifests in the form of dysmetria, intention tremor, dysdiadochokinesia, dysarthria or scanning speech, and rebound phenomenon. Rebound phenomenon refers to the inability to stop a movement of a limb, even the inability to measure the distance correctly for reaching an intended target, which is described as pass pointing. Lesions within the spinal cerebellum leads to diminished muscle tone in the limbs. This manifests as flail joints, disturbances of tendon reflexes, and ataxia. It is very important to note that Romberg's sign is not a test of cerebellar disease and usually refers to a test of the dorsal column. Patients will have issues with balance, whether the eyes are open or closed with cerebellar disease. And that brings us to the end of my presentation.